Proteins are polymers of amino acids. These amino acids are joined by peptide bonds. The amino acids consist of an alpha carbon, which can either be joined to an amino group, a carboxyl group, a hydrogen, or an R group. The R group can either be hydrophobic, polar, charged, acidic, or basic. A protein's function depends on its conformation, which is another word for shape. The protein's conformation depends on a number of structures, which are called primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure is based on the sequence of the amino acids that make up the protein. The primary structure then determines the secondary structure, which is either an alpha helix or a beta pleated sheet. These are created by hydrogen bonds within the polypeptide backbone. The secondary structure then builds to the tertiary structure, which is basically disruptions in charge caused by hydrophobic interactions, van der Waals interactions, hydrogen bonds, ionic bonds, or disulfide bridges. A protein may also have quaternary structure, which is made of several polypeptide subunits.
Secondary structure, tertiary structure, and quaternary structure all determine the function of a protein. The 6 major functions of proteins are support, transport, movement, hormone, defense, and catalyst. When support is the function of a protein, it acts as a fiber. When transport is the function of a protein, it is hemoglobin or membrane proteins. When movement is the function of a protein, it is actin and myosin in the muscles. When hormones are the function of proteins, it is insulin. When defense is the function of a protein, it acts as antibodies. When catalysts are the functions of proteins, they act as enzymes.
Carbon is the basic building block of all life forms. It is essential to evolution and the diversity of life. Because of its valence of 4, carbon is able to covalently bond to 4 other elements. These may bind with functional groups, such as the hydroxyl group, carbonyl group, carboxyl group, amino group, sulfohydrate group, or the phosphate group. These all have specific properties which help make up complex organic molecules. These macromolecules provide for structural and functional diversity within the cell. Carbon may have chains that are either straight or branching, or it may be presented as a ring. These may exist as isomers. There are 3 main types of isomers: structural isomers, geometric isomers, and enantiomers.
There are 6 main types of functional groups. The hydroxyl group consists of an oxygen and a hydrogen covalently bonded to the carbon skeleton. Organic molecules with hydroxyl groups are called alcohols, and their names often end in OL. Carbonyl groups consist of a carbon double bonded to an oxygen. If the carbonyl group is at the end of the carbon skeleton, the compound is called an aldehyde. Otherwise, the compound is called a ketone. A carboxyl group consists of a carbon double bonded to an oxygen and also attached to a hydroxyl group. Compounds with a carboxyl group are called carboxylic acids, or organic acids, because they tend to dissociate to release H+. An amino group consists of a nitrogen atom bonded to 2 hydrogens. Compounds with the amino group, called amines, can act as bases. The nitrogen, with its pair of unshared electrons, can attract a hydrogen ion, becoming negative NH3+. The sulfohydryl group consists of a sulfur atom bonded to a hydrogen. Thiols are compounds containing sulfohydryl groups. A phosphate group is bonded to the carbon skeleton by its oxygen attached to the phosphorus atom that is bonded to 3 other oxygen atoms. The group is an anion due to the dissociation of the hydrogen ions.